So for the flower dissection lab, you're going to get a flower similar to this one pictured here. And you'll see that it has a green stem and some green leaves. And then it will turn into the flower head itself. And the flower head has some parts that you will be removing and looking at more closely. I just want to go through those flower parts with you so you kind of know what you'll be looking at. On the outside of the flower, you're going to peel off what are called sepals. These are sometimes green on flowers, but sometimes they're the same color as the petals. And so sepals are on the outer part of the flower, and when it's a bud, these are surrounding and protecting that developing flower. And then internally, we recognize petals, usually again colorful, and in the case of this flower, they're going to have kind of a stripy slash pattern that's sort of directing the insects to go down those petals into the interior of the flower to find their nectar. So we got petals, and then you're going to see some structures that look like this. I guess I shouldn't have had this upside down, but the dark parts here would actually be standing upright on the flower, and these are known as stamen. Stamen have the little dark part that's shown here, which are called anthers. And anthers are filled with pollen. Now, the pollen has to develop, and when it matures, the pollen comes to the outside and then is ready to be picked up by either the wind, if it's a wind-pollinated plant, or an animal, if it's animal-pollinated. So anthers in the stamen produce pollen. And then if you pull off the petals, the sepals, and all of the stamen, this is what you'll be left with. And so this would be the female part of the flower, called a carpel, although I spelt it wrong, carpel, or pistil is another name for it. And the carpel is made of 3 parts. This top part is called the stigma. And when the carpel is ready to receive pollen, meaning the eggs are mature, then this will become sticky, and so pollen will stick to it. So the sticky stigma. This long skinny part is called the style. And then this part down here is the ovary, which contains the developing egg cells, or a mature egg cell is called an ovum. If you look, you'll have a chance to look at anthers up close, and maybe you can see them in different stages of development depending on what flower you might be able to look at. I haven't checked to see how far along our flowers for this current lab are going to be. So anthers, and then this is be the end of the carpel with the sticky stigma shown. You'll get a chance to look at that. And then you'll also be looking at a cross-section of the ovary and making some assessment of what that looks like. So this is just a snapshot look at what you'll be trying to pick apart as you dissect your flower.